Okay, guys, in preparation for the crime scene lab, I just wanted to briefly go over a few things, to sort of bridge together what we just talked about in Chapter 9 with what we're going to be talking about in Chapter 10. So here's our quick genetics lesson. However, this shouldn't be all that surprising to you. We have two sets of chromosomes in our cells. We get one set from mom, one set from dad. And each set contains the same information, so the same genes. We've talked about this, we've done this before. And we know that each set contains different variations of those genes that we've come to know as alleles, okay? So we can see that here. So mom and dad both have the info for the gene to control eye color. We might call that the allele capital E. Mom says green, dad says blue, so on and so forth. So different versions of a gene are called alleles. And you can see how I've designated them here. Capital E for eye color, but the superscript G for green and the superscript B for blue. We're going to use this sort of way, this way of illustrating this for this lab and for this demo. Different alleles of the same gene produce different proteins. So in other words, these genes, they have the instructions on how to build proteins. We learned that from the celebration. And that those proteins are going to cause this difference in the physical trait. In other words, the gene, this sequence of information in the DNA, is going to give us a protein that produces that color for our eye. In other words, that gene that says green eyes will produce a protein that produces our physical trait. Because we have that protein, our eyes appear a certain color. So that's the premise behind the idea of this quick genetics lesson, is keeping the ideas that the different versions of the gene, these alleles, we get them and they come together in pairs because we have the homologous chromosomes, and that we have these pairs of alleles that give us what our genes are going to say, and what our genes say determine which proteins that we make, and which proteins that we make are going to give us our physical appearance, our traits. Well, red blood cells inside of our body function the same way. They're determined by a gene that has more than two possible alleles. In other words, one allele could say green, one allele could say blue. Those are two possible alleles. But in reality, we know that we could get a brown allele from mom and a blue allele from dad, or a brown allele and a green allele. So I think that as long as you understand that any time that we have more than two alleles that we could inherit, okay, this is going to give us a multiple allele system. But at any given time, we can only ever have two alleles, because we're only going to be able to get one from mom and one from dad. But the potential to have multiple alleles exists. And I know this is sort of probably throwing a few of you, so let's briefly take a look at this idea. Exactly what we were talking about before. Let's say that there's the allele for blue. Let's say that there's the allele for blue. We also know that there could be an allele for green. But let's say there's also the allele for red. Okay? There are three alleles there. But we could inherit a red from mom, a blue from dad. We could inherit a green from dad, a red from mom. We could inherit a blue and a green. Does that make sense, everybody? In other words, the ideas behind this suggest that there are only ever two alleles that are going to express our trait. But we can pull in more than two alleles to make this particular gene in our bodies. Okay, so multiple allele system just means that there are more than two alleles possible. But we can only ever inherit any two of those alleles at any given time because we are diploid and we have two parents. So multiple allele system, this happens a lot. We can get a lot of different possible alleles within our population, but only two can combine to determine that physical trait of our blood. Okay, so I guess the question then is, how does this work in humans. Okay? First, I want to introduce the idea of the genotype and the phenotype. The genotype is based on what our genes say, or the genotype is going to be made up of two alleles. What our genes say, how our genes are programmed, they are based on a combination of two alleles. We get one allele from mom and one allele from dad. So when we refer to our genotype, it's in reference to what do our genes say on our homologous chromosomes. Do I have blue from dad and red from mom? Do I have green from dad and green from mom? Who knows? But the bottom line is our genotype, the types of alleles that make up our genes. Okay? Genotype. Phenotype is the physical characteristics. So think of pheno, think of physical. The phenotype is directly related to the genotype. In other words, our genotype, what our genes say, what the alleles say, determine our phenotype. Because the genotype contains the instructions, but our phenotype is the expression of the proteins. Because remember, we just said that the genes, the alleles, have the instructions on how to build specific proteins. So when those proteins are made and our body expresses them, we call that the phenotype, the physical expression that is based on the genotype. Okay? So keep in mind that a gene is a sequence of DNA that determines which type of protein is going to get produced. That allele, that particular allele, will determine a specific protein that can be produced. Okay? For the ABO blood system, we have 3 different alleles that we can talk about. Okay? And the alleles are IA, IB, and lowercase I. And I always say this, that the gene for the gene here associated with membrane proteins. Okay, so the gene, this I gene, is determining which membrane proteins will exist on our red blood cells. So in the ABO blood system, IA, IB, and lowercase I are 3 alleles for that gene, okay? And that gene determines what kind of membrane proteins are going to be present on the outer cell membranes of our red blood cells. So IA says I have the instructions to produce protein A. IB is the allele that says I have the instructions to produce protein B. And then lowercase I, that we're going to call recessive, says I don't have the proteins. And when you have a lowercase I, no proteins are produced on the red blood cells. So what's interesting is that since we can only ever express a combination of 2 alleles, yet we have 3 alleles to choose from, this gives us a total of 6 different combinations or 6 different genotypes. In other words, these combinations of these 3 alleles in pairs give us 6 potential genotypes. What do our genes say? And remember that the genotype determines the phenotype. And in this case, our phenotype is what type of blood are we? Okay? And from this, we get 4 possible blood types or 4 possible phenotypes. Okay, and this is the easy explanation. So go over this a couple more times. So for example, I might receive the dominant IA from one parent and the recessive from another parent, or I might receive both dominants from both parents. Either way, because I have the dominant I produce A protein, that's going to give me the phenotype of type A blood. In other words, when you have the protein, or when you have the allele that says I can produce A, I can produce A in combination with a lowercase I, or in combination with another dominant I can produce A, you're going to get the blood type A. The same inheritance is true for B. When you inherit the I can produce B protein from one parent, as long as you have that dominant protein, it doesn't matter whether or not it's combined with the recessive or with another dominant protein, you have the ability to produce those proteins B. Thus, your blood type is type B. It is possible for us to inherit a dominant, to inherit the A allele from one parent, and to inherit the B allele from another parent. And if that's true, this means that we have the ability to produce both A and B proteins. Thus, the blood type that we have is type AB. And if we inherit recessive alleles from both of our parents, we do not have the ability to produce proteins. Thus, the resulting phenotype is type what we call type O, and I say type O means no proteins. Type O, no proteins. Okay? So we can get a variety of different phenotypes from the combinations of these different genotypes. Okay, so we can only ever inherit two alleles, but because there are three potential alleles in this multiple allele system, it gives us a variety of genotypes. In fact, there are 6 possible genotypes. That gives us the combination of potentially 4 phenotypes. Type A, type B, type AB, and then of course type O, which means no proteins. Okay, so with this idea in mind, let's quickly take a look at what's inside blood. Blood is really neat because blood is actually a connective tissue that is fluid. It's a connective tissue that connects all of the internal organs and all of the parts of our bodies together. So it connects all of the different systems, all the different parts of our bodies, but it is fluid. So it's a neat little thing because it's a fluid connective tissue. And its job is to carry oxygen from the environment into our blood cells, and then to carry the carbon dioxide from our cells after respiration out through our lungs. On average, 4 to 5 quarts in the body. It's viscous, which just means that it's a thick fluid and it's thicker than water. In reality, blood is made up of 4 components. The plasma, which is a fluid extracellular matrix. The red blood cells themselves, which we know are there to carry oxygen to and from, and to carry CO2 to and from different parts of our bodies. The white blood cells, which we know play an important role in our immune response, and platelets, which help in repairing damage when we get injured. Well, we're more concerned on this lab with these 2 components, the plasma and the red blood cells, because they are going to be what we use to determine what types of blood are at a crime scene. And how we use this is that we're going to actually use our knowledge of what proteins are on the red blood cells and how those proteins interact with the antibodies that are in plasma. Okay, so plasma will give us what we know as antibodies, which attack foreign cells. And the red blood cells, we're going to deal with the proteins, the surface proteins, which are also known as antigens. So we care about these 2 components of the blood because the plasma has antibodies which will attack foreign cells. And then we have the red blood cells that have these surface proteins which are based on the allele systems of IA IB, and lowercase I. So in the ABO blood system, and this is actually really, really cool, if we are type A, in other words, our alleles combine that give us two A alleles, or an A allele and that lowercase I allele, okay, that means that we can produce A surface proteins on our red blood cells. Okay, it'd be like having chocolate sprinkles on a donut. Okay? So remember, this type A is actually represented by I can produce proteins. So we could have that genotype, or we could have the genotype I can produce proteins and I can't. As long as we've inherited one of those dominant alleles, IA, we will produce these A proteins like chocolate sprinkles. When we are type A individuals, we will produce antibodies that will attack type B blood, because our bodies do not run on B blood. We run on A blood because our alleles say so. Our genotype determines that phenotype. Okay? So when we have type A blood, our plasma contains or makes B antibodies, and these B antibodies are going to attack B proteins. But if we're type A, we're good because we have A proteins, our antibodies will not attack our own cells. Well, then we get into type B. Type B, once again, you could inherit two of the B alleles, one from each parent, or you could inherit one of the B alleles and one of the alleles that doesn't produce a protein. But as long as we have one of those B alleles, we have B proteins. This would be like having rainbow sprinkles on your donuts. Okay? So B proteins, and if we produce B proteins, if our genotype, those alleles that come together in our genotype, say that we have B surface proteins on our red blood cells, then we're going to defend against A proteins, so we're going to produce A antibodies. Okay? So hopefully that we are starting to understand is that if we have A antibodies, they are going to attack the A surface proteins. If we have B antibodies, they will attack the B proteins. The idea here, though, is that if different bloods types are mixed together, there's going to be an interaction where the antibodies from one blood type will attack the red blood cell proteins of the other blood type. Now, if we are type AB, it means that we inherited the A allele from one parent, the B allele from another parent. And we produce both A and B surface proteins on our red blood cells, almost like we're having both chocolate and rainbow sprinkles on our donuts. Because we have both types of proteins, in those individuals, their blood plasma has no antibodies. If they made antibodies, it would attack its own red blood cells and it would be dangerous. So individuals with the genotype AB are going to have the phenotype AB blood, and they produce no antibodies. And then, of course, you could have type O blood. And type O means that you have inherited two alleles that do not produce proteins. So there are no sprinkles on this donut, but because it has no surface proteins, the plasma of these individuals will produce both A and B antibodies. So type O individuals will attack any A, any B, or any AB blood it encounters. Type AB individuals will not attack any blood that it encounters because it does not have any antibodies. Type B individuals will attack type A blood, and it will attack type AB blood because of the A antibodies. And a type A individual will attack type B blood or attack type AB blood because of the B antibodies. Well, what does that look like? This is just a fancier way of showing the diagram that I showed you. The idea here being that if you are type A individual, you have antibodies for type B. They are specifically going to latch on to the type B surface proteins. The term that we're going to use when antibodies of one blood type attack surface proteins of another blood type, we're going to call that agglutination, where the cells actually end up sticking together, as you can see here, because the antibodies of one blood type are clamping down on the red blood cells of another blood type, causing all of these cells to stick together. So we end up getting this kind of thing happening when a type A's individuals who have B antibodies attack B blood cells. It's going to happen when a type B individual attacks somebody with A blood cells. Okay? It will occur when the antibodies of a type O individual are going to attack both B, A, and AB blood. Okay? Anytime the antibodies of one blood type attack the surface proteins of the other blood type, the red blood cells are going to stick together and agglutinate. Okay? So what we can do at a crime scene is that we can take the blood samples that we get and we can mix them with the antibodies of type A, type B, and type AB individuals. And upon mixing, we will get what we will consider to be agglutination or sticking together of the cells. If we have a type O individual here mixed with type O antibodies, type O has no surface proteins, type O has both antibodies, but there will not be any sticking together. Okay? If we add A to type O, okay, we will have, or if type O is added to type A, there will be no agglutination. If we add O to type B, there will be no agglutination. And if we add O to type AB, there will be no agglutination because O has no proteins. But if we add type A to a type O individual, the O's antibodies for A will attack the A blood and we will get agglutination. If we add type A to A, we get no agglutination. If we add type A to type B blood, the B has antibodies for A, and it will attack them, and we will get agglutination. And if we add A to AB blood, since AB blood has no antibodies, we will not get agglutination. Type B. Type B added to O. O doesn't just have anti-A, O also has antibodies for B. So the antibodies for B will attack the B blood and we get agglutination. If we add B to A has antibodies for B, so we would get agglutination. Adding B to B, no agglutination because they're the same blood type. If we add B to AB, no agglutination because AB doesn't have any surface or doesn't have any antibodies. And then finally, if we add AB to type O, we know that both the A and the B antibodies will attack it, so we get agglutination. If we add AB to type A, the antibodies in the plasma of A, we'll attack it and we get agglutination. If we add AB to B, the antibodies for A in a type B individuals will attack it. And if we add AB to type AB, there will be no agglutination. So what we're looking for in this lab is evidence of agglutination. We are looking for the sticking together of cells. The reason being, if I add an antibody for type A to a blood sample, and that blood sample agglutinates or sticks together, it means that that blood type has surface proteins for type A blood, meaning that we determine that blood type is type A. If we add B antibodies to a blood sample and it agglutinates, it means that those B antibodies that I added are attacking the B proteins in that sample, thus causing it to agglutinate. Okay, and so it might be hard to see, but you can see what's happening here on this slide, and this is exactly what you guys are going to be doing in the lab, is that we are looking for evidence of agglutination. So this is agglutination, this is agglutination. That's all agglutination. But here you can see that there's no sticking together of anything. It just looks like a blood sample. That's not agglutination. Okay, so this will become more clear when we actually start playing around with this in the lab. But please go back over this animation, over this video, and watch the other videos that are on the crime scene topic, and hopefully this will clear up and give you guys some power before we begin the blood typing lab.